off with a couple talks on the All of Us research program and how to use the All of Us research program. We're then going to go into three concurrent breakout sessions. The first, run by Dr. Chris Lord, is going to be on how to become a researcher in the All of Us program and using the researcher Workbench. The second breakout session is going to be really a deeper dive in some of the tools and analytic strategies. So those of you who maybe played around with the Workbench a little bit and want to learn a little bit more, that would be a great breakout session for you. And the third is going to be two scientific talks from investigators who are using the All of Us research data for addiction research and suicide prevention. And these are going to be by Dr. Hongshang Gui and Jennifer Zhang. So with that, I'd like to introduce our two speakers, Dr. Erica Landis and Dr. Michael Lyons. Dr. Landis is the Scientific and Strategic Partnerships Advisor for the All of Us research program, and she supports the ancillary studies and the scientific partnerships that advance the innovative research of the program. She earned her Ph.D. in neuroscience from Emory University and has led the development of collaborative research programs across a wide range of different scientific fields. She has expertise in strategic planning, inclusive communication, and implementing evidence-based policy. In the All of Us program, Dr. Landis aims to advance partnerships that strengthen the All of Us program as a resource for researchers. Dr. Michael Lyons is a scientific project manager for Vanderbilt University Medical Center and is a member of the outreach team for the All of Us program's Data and Research Center. He focuses on helping researchers use the diverse resources that the All of Us research program contains and how to really customize them for their own research needs. And with that, I'm going to give you Dr. Landis and Dr. Lyons.
Thank you, and good afternoon, everyone, or good morning, depending on where you are. We can go to the next slide. I'm going to give a quick background on the program. So starting off in January of 2015, President Obama launched a new U.S. initiative called the Precision Medicine Initiative. This was designed to accelerate our understanding of individual variability and their effects on disease onset, progression, prevention, and treatment. The next year, the government dedicated funding to start the Precision Medicine Research Cohort, which soon became the All of Us Research Program. President Obama said about this effort, "My hope is that this becomes the foundation, the architecture, whereby 10 years from now, we can look back and say that we have revolutionized medicine." Today, All of Us is part of a new era in which researchers, healthcare providers, technology experts, community partners, and the public can work together to develop individualized healthcare and revolutionize medicine. Next slide. All of Us is a precision medicine program of the National Institutes of Health. Our mission is to accelerate health research and medical breakthroughs to enable individualized prevention, treatment, and care for all of us. We do this in three ways. The first is to nurture partnerships with our cohort of soon to be 1 million participants who reflect the diversity of the United States. We also catalyze an ecosystem of communities, researchers, and funders to integrate all of us into regular health research and deliver the largest, richest data set broadly available, and securely available for research. And you'll be hearing a lot about this today. Next slide. Under our current protocol, anyone living in the United States who is 18 years or older and able to consent on their own can choose to enroll in All of Us as a participant. We welcome participants of all health statuses and are working to make the program available in the future to children, people with decisional impairments, and incarcerated individuals. When participants first enroll in the program, they consent. They're given the option to authorize sharing of their electronic health records with the program. Any electronic health record data that's shared is cleaned and curated and de-identified before being made available to researchers. Participants are invited to complete surveys at their convenience, and I'll talk a bit more about what each of these surveys are in a second. They also agree to share After they agree to share their electronic health records and complete at least one survey, they receive an invitation to share physical measurements with the program. They may also be invited to share biospecimens, blood, urine, and/or saliva with the program from their enrollment center, a designated collection site, or through an at-home kit. And finally, participants can opt to share data from Fitbits on an ongoing basis. And I'll dive a little bit into each of these. Next slide. We are currently collecting electronic health records from participants located in healthcare organizations, as well as direct volunteers who sign up online. The initial data collection through electronic health records includes demographics, clinic visits, diagnoses, procedures, medications, laboratory visits, and vital signs. We recognize that there's a lot more information in healthcare records, and so we're working to expand this list to include other data types like clinical notes, radiology notes, mental health reports, more details on substance use, alcohol use, and tobacco use. You'll hear about what we do have today, but we would like to expand this. And more laboratory results, potentially including clinical genomics testing and images. Next slide. In addition to sharing electronic health records, participants are invited to respond to surveys about their healthcare access, family medical history, personal health history, and more. You can see the list of open surveys on the slide here. We have the basic survey, overall health, lifestyle, healthcare access and utilization, personal and family health history, social determinants of health, and two relatively new surveys that were released in July, emotional health history and well-being, and behavioral health and personality. We also have data related from two surveys that are now closed. So we're no longer collecting data from those surveys, but the data that was collected is available to researchers. These are both COVID related, the COVID-19 Participant Experience Survey and the Minute Survey on COVID-19 vaccines. We also plan to release more surveys on an ongoing basis, and that data will become available to researchers when it has been cleaned and released into our Workbench. Next slide. For physical measurements, we currently collect data on blood pressure, heart rate, height, weight, BMI is calculated, hip circumference, and waist circumference. Participants are given access to this information through our participant portal, and if they have those measurements done in a clinic, they're given a physical card when they leave their visit. Next slide. Biosamples are shared with the program either from the participants' enrollment center, they're a designated collection site, or in the case of saliva, an at-home kit. These samples are currently banked for future analyses, either through the program or through ancillary studies. While the program currently has several hundred clinics open around the country, we don't have sites next to every participant, which is why we give several different options for sharing biosamples. And we do try to make sure to extend invitations to a broad range of participants so that we make sure we have a diverse cohort of individuals, for example, those in urban and rural communities. Next slide. Through the program, participants are also able to share data from their Fitbits. If a participant already owns a Fitbit, they can share that through our Bring Your Own Device project, or invited participants from communities previously underrepresented in biomedical research may be invited to our WHERE study, where they receive a free Fitbit and can send the data through the program that way. Participants also can link their Apple Health Kit data. And we're working to expand this to include other wearable devices and apps so that participants can share different types of data from different sources, including related to their mood or cardiorespiratory fitness. Next slide. Oh, one more. Thank you. The program has established 3 genome centers around the country to generate genomic data from the biosamples donated by participants. And you can see the list of organizations involved in these genome centers on the right. As a part of this effort, we've also partnered with Color, a health technology company, to establish the program's nationwide genetic counseling resource for participants. They provide educational materials, telecounseling in multiple languages, and resources so that participants, as they get back their genetic data, can have effective conversations with their providers and better understand their results. Next slide. The program is able to provide some genetic information back to our participants, and providing information to our participants is a goal of the program for all of the data that we collect. So we are able to provide this information, engage them in research, and provide some value for their participation. Through a genetics engagement module, they can access information about their genetic ancestry and traits like food preferences, and they can choose to access genetic counseling if they like. We also are currently providing 2 reports related to genetic results that participants can consent to receive. This includes the Hereditary Disease Risk Report, informed by recommendations from the American College of Medical Genetics and Genomics, which includes 59 genes and variants that participants can get information about related to serious medically actionable health conditions. And the Medicine in Your DNA report, which includes 7 genes that are known to affect how the body processes certain medicines. Over time, we hope to provide even more genetic results and other types of results related to the other data that we collect to our participants. Next slide. So before I turn it over to Dr. Lyons to talk a little bit about what data is available to researchers through the program, I want to express our deepest appreciation for the All of Us participants. None of this would be possible without them, and it's only through their generosity and the involvement of our researchers that we're able to create these resources and drive precision medicine forward. So now I will turn it over to Dr. Lyons to take a deeper dive into the data available to researchers through the program. Thank you.
Great. Thank you, Dr. Landis, for that great overview of the program. I'm going to kind of jump into a little bit more details on what the data set is, what our goals are, how we're approaching those goals, and how to make this data set useful for you as researchers and how you can engage with it. So if you can go to the next slide. So as Dr. Landis mentioned, the goal of this program is to develop a diverse biomedical database with up to and beyond 1 million people across the United States. So we currently have over 400,000 participants data that is ready for researchers to work with and engage with. And on the focus of making that a diverse biomedical data set, we're very proud to say that 75% of these participants come from communities underrepresented in biomedical research, and 45% specifically are from racial and ethnic minority groups. So next slide. So really the idea of this data set is to have a very diverse data set that can influence personalized medical approaches in the future. So this is aimed at combining a number of different data types from biological factors and social determinants, having a longitudinal data set that follows participants as they age, as they move to different areas. And we want to make this accessible for researchers to really engage with and continue to work with this data set over time as it grows and answer all these different potential questions that can be addressed with our data set. Next slide. So what really makes this data set so special is the fact that it is kind of overrepresents underrepresented participants that have historically been underrepresented in biomedical research. So you can kind of see some of the breakdowns here. So as I mentioned, about 3/4 of the participants come from at least one category of underrepresented biomedical research categories, with 43% coming from a non-white race or Hispanic Latino ethnicity. Almost 1/4 of them are older than 65 age. Almost 10% have less than a high school education degree. 1/4 of them have an annual income below $25,000. 10% are sexual and gender minorities, and 10% have a disability. So this really allows researchers that engage with this data set to answer and study participants that have really been out of reach for a lot of different major studies in data sets like these before. So next slide, please. And if we actually deep dive into what these numbers are for our specific data types, you can find that here. So as Dr. Landis mentioned, we have a number of different data types, including survey responses, physical measurements, genomic data in the form of arrays, whole genome sequences, structural variants, and long read sequences are new as of this April. And then we also have electronic records and Fitbit records as well. So as you can see, over 400,000 participants have filled out survey responses, and that's really the first step of becoming in all of us participants is filling out these basic surveys, and from there you can add on to these other data types. So we're continuing to have these data types grow with the number of participants, as well as expanding the data types that are offered. So we're very excited about that, and really the power of this research, if you go to the next slide, is not just the fact that we have all these different data types, but the fact that you're able to combine these different data types for a large number of participants. So currently, the All of Us Researcher Workbench has the largest set of whole genome sequences available in a data set like this, which we're very, very proud of. And that's over 245,000 participants with whole genome sequences. What's equally amazing is that for over 200,000 of these participants, you're able to link that genomic data to data from their electronic health records, from their physical measurements, and from their survey responses that capture different social and economic factors. So it's a really, really rich and deep data set that you can really engage with and match all these different aspects of influences of a participant on their healthcare and life. So if we go to the next slide. As Dr. Landis gave an overview of these different surveys that participants are able to fill out. So I just wanted to highlight one that might be of particular interest to this group. So if we go to the next slide. One of our more recent surveys is the Social Determinants of Health survey. And this is a number of questions to the participants that cover a range of kind of environmental and personal aspects of that participant's life, such as their neighborhood safety and environment, their housing and food security, stress, loneliness, discrimination. So really try and get a holistic view of these different factors that are impacting our participants and their life and their health as well. So this is a really great resource to combine with different things such as electronic health record data, genomic data, to see how different outcomes are caused. So if we move to the next slide. What's also wonderful is that we really are getting a lot of researchers engaged with this data set, because the goal of this data is to be used widely and wisely by the researcher community. We're happy to say that over 590 institutions are signed as partners with the All of Us program. We have over 7,250 researchers that are registered, with over 7,000 research projects ongoing. And there's a wide range of researchers and interests that are using this program's data sets, from bioinformatics, bioinformaticians, that's a hard word to say, data scientists, epidemiologists, geneticists, and a lot of them are early career researchers and students, and a lot of people are also looking at health equity and health disparities. So there's a lot of different ways to engage with this, and we're really happy to see a lot of researchers such as yourselves are engaging with this resource. So next slide. So to kind of cap that off, how can this really be applied to your research, and how has this been used previously? So I kind of wanted to touch on that briefly. So as many of you are likely aware, the drug overdose death rates have gone up over the past 2 decades, especially if we look at the past 10 years of opioid death overdoses, as you can see on the graph on the right, compared to all drug overdose deaths on the left. So this is a really major thing that we need to research and understand more about. So if you go to the next slide, we have had researchers that have engaged with the All of Us research program dataset to see if this is a resource that we can use to understand kind of different relationships and causalities of these opioid crisis and death overdoses. So if we go to the next slide, we can look at a specific table from that publication. And this really highlights the power and what resources are available with this data set. So on the top of your screen, on the top kind of x-axis, you can see all the different data types that are used and are available that are related to drug use, substance use, and opioid use specifically. So we have different survey data type. We have data from electronic health records, and then we can further go on and look at how the different demographics of our participants and how that relates to their responses in survey data and what is found in their electronic health records. So overall, the All of Us data set really provides a up-to-date and more diverse data set than compared to other large data sets such as NSDUH and NESR. SARC to look at this type of data. So it's a really exciting resource to really jump into and utilize. Next slide, please. So next, I'm going to be talking about how researchers can engage with this data and how we make that available to you. So next slide. So essentially, the way that we get data to researchers is that it goes through a research data pipeline. So the Data Research Center, or DRC, is a part of the All of Us program. And we take in all these different data sources from electronic health records, genomic information, surveys. We harmonize that data and refine it and develop a curated data repository. So all the data that you get and receive on the researcher side is this curated data, and we make that then available to our researchers. So if you go to the next slide. There are a number of different ways to engage with this data. So we break this data down into 3 tiers. We have the public tier on the far left, which is available to anyone. And this has different aggregate counts, rounded to nearest 20. So this is a great resource to look at to see, does the All of Us program have the data that I'm interested in pursuing for my research project? Because while it's a very vast resource, it's not all encompassing. So that's a great place to kind of see, do I want to engage with this? Do I want to pursue this? And if you say yes, they do have that data, which hopefully we do, then you would become a registered user and get access to the registered tier and potentially control tier as well. On this aspect of the registered tier and control tier, this data is available on our researcher Workbench, which is our cloud computing platform. And this is where you'll actually engage with the row level data, as well as kind of learn how to build your cohorts, your data sets, and then work within Jupyter Notebooks to analyze said data. So if we go to the next slide, kind of giving a little bit of a breakdown of what's difference between the registered tier and controlled tier. Registered tier has a lot of the data. It does not have some of the more unique and row level and deep dive data, such as genomics and health and lifestyle survey data. But it does have surveys, electronic health records, physical measurements, wearables. Some of the dates for things in the registered tier are shifted within a year, while in controlled tier, you get the exact dates of events. And the difference between kind of getting registered tier versus controlled tier depends on the agreement that your institution has with the All of Us program. So if your institution has an agreement for the controlled tier, then you would be able to go through the training to get controlled tier. And it's not a big hurdle versus the registered tier. So if we go to the next slide. I'm going to go into the Research Hub and give a slight overview of that. And this is really our public facing tool. So you can see the Research Hub here. We have the URL there. And this is a great tool to explore and kind of learn more about our data set. So we have data snapshots, and that's where you can see where we currently are progressing for reaching that goal of 1 million. What does our participant demographic breakdown look like, from geography to ethnicity, male versus female. So you can really jump in there and see what the overall data set is looking like currently. If we go to the next slide, another incredibly useful tool, and what I highly recommend to kind of start forming your hypotheses and seeing if we have that data available, is our data browser. So as you can see on the screen, this data browser kind of lists all the different data types that we have available for participants. So we have our different EHR domains, labeled in conditions, drug exposures, lab measurements. We have measurement wearables. We have our survey questions where you can go and see what the questions are, how many participants have filled out those specific surveys. And we also have a really cool tool with our genomics data. So if we go to the next slide, what you can actually do is you can check out and search for genomic variants to see what SNPs and variants we have available within our All of Us data set. There is a huge amount of SNP variants, as you can see, over 1 billion different SNPs from across almost 250,000 participants. And since this is a very diverse data set, there are a lot of unique SNPs that haven't really been characterized before and really studied. So if you're interested in genomic research, highly recommend checking that out as well. So if we continue, you can also identify what other research projects are ongoing. So to make this very transparent and to make sure that the research and data set is having a positive impact and not stigmatizing, all of our researchers' projects descriptions are made public on the research browser. So this, you can go in, you can find what research is ongoing in a different specific area of interest. You can find potential collaborators. It's a really great resource to look at as well. So next slide. And then very finally, if you do become a registered user, which we all highly recommend, and decide to engage with this data set for analysis, you'll move on to the Researcher Workbench, which is our cloud computing platform. So if you go to the next slide, this is essentially the home page that you'll land on. And there definitely is a little bit of a learning curve for getting used to this, but we will have a deeper dive in one of our breakout sessions to kind of get you all oriented. But it's a great resource to go in, start working with the row level data, and code and analyze this with either R or Python. So that's the overview of everything. So I think we will start moving into the breakout sessions. So I think you go to the next slide, and I'll hand it off from there. And thanks for coming in and listening.
Thank you so much, Dr. Landis and Dr. Lyons. I really appreciate your overviews of the program. And with that, we're going to open up the breakout session shortly. Please pick with a breakout session you think you'd like to enter. And just as some housekeeping, we are recording the entire webinar, so that will be available to you guys. And we are going to be keeping questions in the breakout sessions to the end. So with that, we'll see you in the breakout groups.
Okay, perfect. Thank you everyone for your patience. We will start session one. Next slide, please. All right, so this is just a disclaimer. Any data that we might show is not our actual data. We do have a user policy where you can only be a registered user to see our row level data. Therefore, we are showing you some operational synthetic data for any demonstrations. And we'll hold all questions until the end of the section. Next slide. I would like to introduce our speaker, Chris Lord. Dr. Lord is a project manager here at Vanderbilt University Medical Center, and he primarily assists with research support for the Data and Research Center, also known as the DRC. He focuses on user support, on the User Support Hub, our featured workspaces, and supporting materials for users. So welcome, Dr. Lord. I'll turn it over to you.
Thanks, Danielle. Hopefully everyone can hear me. This is one of the shorter ones, which I think we can get through in our 30-minute allotted period. I'm going to give a quick overview of what I'll be talking about. I'm going to give an introduction to what the Workbench is, to sort of follow up on what Michael discussed, and give some information about how to sign up for the Workbench. Also, I'll demonstrate how we use workspaces to organize scientific projects, how you can collaborate on the Workbench by sharing your workspace, two tools that we have that are really useful for people, especially beginners, like our cohort and dataset builders, how you can utilize Jupyter Notebooks to analyze your data, and then provide some information about credits, which I think is useful to learn for new users. And then finally, I'll quickly wrap up with some information about how to find support resources. We realize the Workbench is a complicated tool, so we've got a lot of support materials ready, including featured workspaces, our user support hub, and help desk and office hours for one-on-one support, to sort of guide you through that process, because it can be kind of daunting, especially for new users. So, what is the Researcher Workbench? It's a cloud-based platform that's used to analyze participant-level all of us data. And just to sort of define some of those terms in a way that I think hopefully makes sense to everyone here. When I say participant-level data, I mean that means you can analyze different variables together at the same time for individual de-identified participants. So Michael was describing the data browser, which is a wonderful tool, and it's publicly available. There you'll see aggregate data. So for example, if you wanted to know the number of participants with type 1 diabetes, you could easily find that total number in the data browser. You would not be able to further analyze those individual participants in any way and find out what else, what other types of variables or phenotypes they have associated with them. In order to do that, you actually have to use the Workbench. And so that's what, and so in the Workbench, if you know one condition about someone or one type of variable about someone, you can analyze a different one at the same time. And by cloud platforms, in case people aren't familiar with using them, which I wasn't until I joined the DRC, it's when you use virtual machines that are accessible through the internet to analyze data in some way. I think mostly cloud platforms offer advantages, but just in the sake of transparency, I want to mention some that I think can be disadvantages. So I think especially for people who have not used it before, I'd say there's a reasonable learning curve, which happens to everyone who's basically moving onto the cloud. So if you have cloud experience, you might not have that issue, but if you're new to it, my guess is you'll probably need to read a decent amount about it to learn about how to actually, do your analyses in the cloud. I also would say troubleshooting can be difficult at times, and that's because if there's some kind of technical problem, unlike a local machine where you can maybe troubleshoot it to some degree, you can't do as much, and you might need to contact our support team, which I'm a part of, and we're happy to help for assistance, but that might take a little bit longer when you encounter those errors. Now, for the advantages, which, like I said, I think generally outweigh the negatives, and one specifically I'll talk about at the end. So the nice thing is wherever you have internet access, you can access the Workbench and other cloud platforms. And since the internet is basically ubiquitous now, it's basically anywhere, right? At least in the US. It's easy to collaborate in the cloud, and I'll show you how to do that in Workbench. You can easily customize your environment, which is nice. So if you need a lot of power one day for a type of analysis, you can use that, and then you can change it back down so it's much cheaper the next day for something that doesn't require as much. There's also less initial costs because you don't need to necessarily buy, large-scale equipment to do your analyses locally. And then the main reason that it's good, especially for the All of Us program, is that in this case, it protects participant privacy. That way, people aren't having, de-identified data sets on their own local machines that could be easily shared. And it's basically, the data is on the cloud, and the main advantage for the All of Us program is that it's the best way to protect participant privacy. So that's sort of, I just have a few slides, and this is actually the last one. I'm going to mostly just do a demo to sort of show you how to sign up and use the Workbench as a new user. The way you get started is by going to register for the Workbench at researchallofus.org/register. So that was that site that Michael talked about. And so I should have it pulled up here. This is the Research Hub. As Michael mentioned, you can find the data browser and other types of really useful tools and information about the Workbench and dataset from the perspective of a researcher, right? But if you want to register, you'll see this, this orange button up in the right corner. Click it, and there's basically 4 main steps to register to become a Workbench user. The first of which is probably the most important, and it's not in your control. So you have to belong to a healthcare institute, nonprofit institute, or academic institute that has signed a data use and registration agreement, or DURA, with the All of Us program. And I'll talk about that in a second. Next, you'll have to verify your identity using login.gov, complete some training steps, and sign a data user code of conduct. Steps 3 and 4 are really easy. Step 2 can be a little bit of a problem depending on some login.gov issues that we have workarounds for that. But the first step is kind of out of your control, but the last 3 are pretty much completely in your control, and I'll go through them in a little bit more detail. So the first thing you want to do is actually check whether or not your institution has one of these agreements. Now, the nice thing is, I think we're at, so we're almost at 600 institutions now in the US that have this agreement. So odds are you're probably associated with one of them that does. And you can easily check this. We have a list here, but you can also just quick search it. So I'm associated with Vanderbilt Medical. If I just put in Vanderbilt, I see that VUMC has access to registered tier and controlled tier data, which is great. So that means I can move on to the next step to apply for access. What happens if you, and I would do that by clicking create an account. I'll do that in a second. But what if you are belonging to an institution that you think is eligible but doesn't have access? In that case, you can click this button here to submit a form, which will, you can provide some information about your institution. And our access team will reach out to them specifically to try to get them to, sign one of these DUR agreements so that you can then access the data. So those are kind of the two options you can get. We're going to assume you have access. We'll go to create an account. That'll take you to the Workbench. You can then click create an account. Obviously read the terms of use better than I did. I'm not going to actually make one though. Sort of again, going along with the fact you need to be associated with an institution that has a DURA. All those institutions that I just showed you on the researchallofus.org page are here. And so again, I'll put Vanderbilt, that comes up, and I can put in email. And, as long as you get this green check mark, you're good to go. And then you can basically start going and filling out some other information that I'm not going to walk through now because it's pretty straightforward and will take a while, but you basically click the next button and it'll sort of get you to the page where you have to do steps 2 through 4. So I'm going to go there. And so after you completed those initial steps, you'll end up on this data access requirements page. You'll have to activate Google two-step verification. And then, as I mentioned, the next step can be tricky, especially if you're not originally from the US and you come here recently, where you have to verify your identity using login.gov. I will say that if you can do it, that's great. Do that, it's faster. But if you have any issues, you can always, and I'll talk about this again at the end, you can always click this button here and click contact us and then submit a help desk ticket. And we have a bypass system in place that does take a while. But we'll be able to get you verified in a different way so you can actually start using the data. That's really the main difficult step on your end. Next, you'll then need to complete AOU research registered tier training. Once you've done that, you're almost done with the requirements, and then you can sign the data user code of conduct, which just sort of you agree to basically not share any participant level data, among other things. You can obviously read that for yourself when you're ready. Now, this just gives you access to registered tier data. If your institution has also signed a controlled tier rider to give their users access to controlled tier data, i.e. genomic data, you can fill this out too if these are green. I generally recommend if you have the ability to access controlled tier data, you would just do that training and have access to it because there's no real downside, except, you have more data as opposed to less. But anyway, so this is optional. It's not actually required. Can do it, and I'd encourage you to if you can. Okay, so next I'm going to go to the main homepage of the Workbench. So after you do these, right, you can click Get Started. It'll take you to our main page. The main thing you'll see is a list of workspaces that you may have. And then you'll also see a quick tour and videos, as well as any data sets you may have recently accessed. And so the videos are down here at the bottom. You can always view these. And also there's some nice quick links at the bottom to the Research Hub and User Support Hub, which I'll talk about this User Support Hub at the end. Basically, I want to just briefly go over how to use the Workbench. I'm not going to go in depth, but we have a lot of video tutorials and documentation about how to do so. But just to give you an idea of what you can actually do on the Workbench. So generally, scientific projects are organized into workspaces, where one scientific project corresponds to one workspace. To give you an idea of what that kind of looks like, I'm just using this demonstration workspace I set up. You'll notice 3 tabs at the top: Data, Analysis, and About. I'll start with the About section. So when you create a workspace, you'll be filling out information about its research purpose that will be available on that public researchallofus.org directory that Michael mentioned in his first part of your talks here today. So basically, you want to be sort of thoughtful about how you fill these out. That said, you can always change and edit these at any point. So we realize that it's likely your research topic might change as you're doing something. That's completely fine. You can always update it by clicking these buttons here and clicking Edit Workspace Actions. I mentioned that it's easy to collaborate on the cloud, specifically the Workbench, and that's true. And one nice thing is, so as long as you want to share your work and your workspace with anyone else who has the same level of access that you do, I registered a control tier, you can do so by clicking the share button. And so, for example, if I want to find my colleague Danielle, I can just type in her researchallofus.org address, click this plus button, and then give her different types of access depending on what I'd like to provide. So for example, if I give her owner access where she can add other people if she wants, I can click the save button, and you'll see in probably about 10 to 15 seconds, once this loads, she'll be listed as an owner right here too. So you see now that Danielle can also have access to this workspace if she wants to. So this is one thing I really like to highlight, because it is very easy to work together in the Workbench, and I think it's one of the strengths of the platform. Especially, if you're with someone who's at a different institute than you. I'm now going to move so Danielle mentioned that I'll be showing, I probably will be showing row level data. I'm going to move to a different environment that's used for demonstration purposes called Stable. Functionally, it's exactly the same as the main researcher Workbench, but it has synthetic data. And you can see here it's listed as stable and stable. So if you see any data, don't worry. It's not actual participant data, it's just sample data. But I want to do this to show you what it looks like to actually use the two tools that I mentioned, the cohort and dataset builders, which allow you to sort of use user-friendly UI tools to select a group of participants from the larger data set, which is, I think at least almost, it's like, 425,000 people, and with the cohort builder. And then with the dataset builder, actually pick different variables about your cohort or concept sets that you want to actually analyze in data set with a specific cohort. So I'll start with the cohort builder. So let's say, for example, I want to look at the survey responses of participants who are between the ages of 40 and 80, and also have short read whole genome sequencing data, for example. So what I can do is I can use these nice friendly UX tools. So I can go to demographics and click age. And for example, if I want to select people between 40 and 80, I can just literally put this number in. And then you'll see it gives me about 170,000, like I said, fake participants here. And then I can add the selection and save it as a criteria. And the nice thing is it'll give you the list of people that actually, fit with the data you're wanting, right? And then you can use Boolean and or logic to actually, make it more precise depending on what you want. So I'm going to use and criteria because I want people who also have short whole genome sequencing data, right? Actually, I'm going to pick something else because since this is fake data, we don't actually have short read whole genome sequencing. So let's pick people that who had a sex assigned at birth of male as a designation. And the nice thing is you can delete these. So again, there's 87,000 fake participants here who, are contain sex male assigned at birth code. You'll notice that, since this is an AND logic, the total count is actually lower than both. You can also exclude criteria. So if for some reason you wanted to exclude specific people, you could do that. Since I'm looking at survey data, I'm actually going to make sure they've actually completed this COVID experience survey. So I'll click finish and review. And basically that'll be a way to actually create my cohort, right? And I can save it, which is nice. So we'll put NIDA demo cohort. Click the save button. And then basically there I have my cohort that I want to actually analyze. But let's say I picked that cohort. I wouldn't actually have anything other than their person IDs associated with them. In order to actually pull specific types of data that I want, in this case, survey questions, I do so in the dataset builder. So I can do that by, you'll notice the United Demo cohort is saved here. I'll do this by going to the COPE survey that I'm interested in. And the question I'm interested in specifically is, let's see, I want to see if, or actually, wait, it's a COVID-19 vaccine survey, excuse me. Looking in the COPE survey, which is a different COVID one. I want to actually see whether or not the people who responded when they responded to the survey thought they had COVID-19, right? So I'll click that as an option, click Finish and Review, and save that concept set. COPE survey. And basically what I would then do to make a data set is I would select this cohort that I created, right? And then select the different types of concept sets or variables that I want to pull from them. And so in this case, I just want to pull COPE survey data in that one question that I'm interested in. We have some pre-made concept sets. If you, like, for example, a lot of the Fitbit data isn't so easy to find. So we have these, these pre-generated ones here. Different surveys if you want them, and demographic data. Generally, I recommend pulling in the least amount of data that you need to do your analysis so that it's less expensive. But that's just sort of a general tip. You don't have to actually follow that. Another thing you can do is before you actually pull a data into Analyze, or data set into Analyze, you can view the preview table, which gives you sort of a row level indication and demonstration of what your data will actually look like in a Jupyter Notebook for analysis, right? And so generally, again, I recommend people check this out before they actually make something. You would then pull the data set into a notebook that I'm going to make here. And if someone asked, we have Python or R as options right now. I'm going to just select Python by default, and then export this data set to a notebook, right? And basically what will happen is it'll create a SQL query that will just, when you run this, will actually pull the data that you want automatically, which is really nice. Just for the sake of time, I'm not going to actually start up an environment and show you what this looks like in practice, but this is sort of where the cloud part comes in. So we utilize Jupyter Notebooks, which are sort of a cloud-based tool to analyze data that are powered by VMs. And so what I would do is if I actually wanted to run this, it would take a few minutes to get set up, which is not uncommon for cloud platforms, by clicking the Edit or Run button. And then it would create an environment with different options that I have pre-selected or that I customize myself. So basically, in order to actually run an analysis, you have to have a cloud analysis environment set up first. We have some recommended environments that are, we have general analysis VMs, which have, you can increase or decrease the amount of default CPUs and RAM they have. You'll notice that when you're doing this, it lists the cost per hour to run your environment, and that'll change automatically as you change the settings, right? And you'll obviously notice that as my VMs grow in power, the cost while running increases. You can also use data clusters for Hill genomics analysis if you want to do so. You can select the type of short-term disk storage you want and the size. Basically, there are a lot of options to customize your environment depending on what you want. Again, ideally, you choose the smallest environment that will work for your research, but if you need a large environment, it's there. That sort of leads me to the last thing I want to mention here is that when you start and you sign up on the Researcher Workbench, you get $300 in free credits to compute your data. And the nice thing is, even if you signed up, 3 years ago and you come back, as long as you haven't actually used them for anything, they'll still be there. So they never expire unless they're actually used for, actual analysis. I'm happy to go into more details, but generally costs are paying for your environment when running. The amount of data that you actually pull in to analyze from Google BigQuery, which is where the data is stored. And then also any types of associated costs with storage. Generally, the storage costs are much lower than environmental and data pulling costs, in case anyone's interested. One thing I want to mention too is that just having a workspace and using the cohort and dataset builders is completely free. So if for some reason you're actually just wanting to get, learn about how to use the Workbench and use these tools without actually using your credits, you can do so, which is kind of nice. One thing I want to mention is that this is, these tools are generally recommended for non-genomic data, that we have an extraction tool for smaller cohorts that will work for genomic data. I'll talk about this a little bit, but if you actually want to analyze genomic data in the Workbench, you generally have to pull it yourself while you're already having an environment set up, which we have information for. And that sort of leads me to the last thing I wanted to talk about, which is, support services for when you have questions or you want to learn how to use the Workbench and analyze its data. So I'm going to close this and go back to the normal production version of the Workbench. The thing I'd encourage people to look at first are featured workspaces, which are in this main menu here. We have different types of featured workspaces that demonstrate how to actually use the Workbench and analyze different data on it, and also provide examples of how the data has been used in demonstration projects and phenotype libraries. For new people, I would encourage you to check out this first one that's listed. Basically, it was created by Imon, who's one of the other people who's giving a presentation now. And essentially, it provides notebooks that allow you to interactively walk through how to learn how to analyze data on the Workbench. So especially if you're not familiar with cloud environments and or the All of Us data set and how it's organized using OMOP, I would encourage everyone to check that out because this is a fantastic resource. If you're curious about different types of data, for example, COPE survey data or physical measurement data, we have a featured workspace, and I mentioned genomic data. So for those of you who want to work with it and are trying to pull it in yourselves without using these tools necessarily, we have, Jennifer, the other, one of the other leaders today in the third session, created this featured workspace, which basically guides you through how to work with the genomic data in many ways, not every way because that'd be impossible, and even to the point where you have a walkthrough about how to actually do a GWAS if you're interested, right? So I'd encourage everyone to start here if they have questions and want to learn about how the data is used. Next, I'd suggest going to the User Support Hub, which you can find just in the main menu of the Workbench. And so this is our support page that lists documentation and just resources for learning about how to use the Workbench in the data set. So we have a Getting Started section with just general information about the program and the Workbench, videos where we demonstrate how to do different things, different types of sections depending on what you're looking at. You can always go to the main search bar and find something. So if I want to learn about OMOP, right, I can just type that in and find a bunch of articles that are associated with that topic. So if you have another question you can't answer that's not answered by the featured workspaces, I'd recommend coming here, but you obviously don't have to. And that, if you do have further questions after coming here, you wanted to skip directly to this step, you can always contact our help desk by clicking this help button, and that's on the support hub, by clicking ask a question or reporting an issue like you would for login.gov issues that I mentioned. Or you can do so again on the researcher workbench if you're just there and you want to ask a question, right? You can contact us, provide your email address, and we'll get back to you within 1 to 2 business days. The last thing I want to mention is that if you, we do have a lot of great live events that we do weekly, if not monthly, depending on what they are. So for registered users, we send out a link that has where we provide drop-in office hours where you can come in with either pre-submitted questions or questions that you just want answered quickly to help you with your work, and we do those weekly. We have a New Year's orientation, which is kind of similar to what I talked about today, except a little bit longer, every month. And then we also have office hours every 2 weeks on Fridays, where we provide sort of general topics about how to use the Workbench. And in 2 weeks, there'll be an intro to working with the HR data by someone on our research support team that I think will be really useful. You can always find previous recorded versions on our YouTube page or on the support hub, in case you're wondering. But if you want to find this calendar, it's publicly available on our support site just at the top here. I think that's mostly it. I'm happy to answer questions now, but basically I just wanted to emphasize that the Workbench is a really powerful tool that I think will help you get started with analyzing the Workbench. As I mentioned, it can be complicated, and we realize this, so we have a lot of resources that are available for people, particularly new users, to sort of help guide them through the process. So I'll stop there. If anyone has any questions, I and my colleagues are happy to answer.
Feel free to come off mute and ask your question.
I have a question.
So I'm just wondering if it is possible to construct a disease cohort, for example, like a HIV, using the diagnosis kind of ICD-9, ICD-10 code.
Yes, there is, and you can do that. You can. Well, another thing I actually think I overlooked, which was my fault, You don't actually need to use the cohort dataset builder, so you could pull this data in yourself in an active environment using SQL if you wanted to. But the general answer is yes, and you can do so by, I'll go back, actually, I'm going to go back to stable again just to try not to do something incorrectly here. But yeah, you can basically search in the cohort and dataset builders using that as, like, for example, HIV as an option, right? So as a criteria. So I can put in HIV, search that, and you'll see that basically this, this don't, like, the HIV comes up in different types of data that's associated with people, right? So if you want conditions, you probably want to go there. But also, obviously, that information is included in some surveys that people fill out, and obviously related labs. But you could go to conditions, for example, and then actually find the concept ID you're looking for, and then add that to your cohort, if that helps.
Oh, that's great. Thank you.
Yeah.
Michael, I see your hand raised.
Yes, thank you very much. I wanted to know, is the access to all of us restricted to only people from the US and the UK? Because I was working with a lab and I tried assessing it for education purposes, and I couldn't access it. I'm from Nigeria, so I wanted to know if it's still possible for me to access it for educational purpose.
Yeah. So if you're an international researcher associated with a U.S. based healthcare institute, not for profit or academic institute, you can. But and I will say that we don't have JIRAs in place yet, though they probably will be in the next few months for some international academic, not-for-profit healthcare institutions, and you can apply for these. So, for example, Michael, you might want to submit a request on behalf of your institute that you're at. We're accepting applications for international JIRAs. So it's possible that while you don't have access now, you might in the future. So for Michael, I'd recommend going to this, and actually, I can pop. Oh, great, Cassie put the request form. Thank you, Cassie. If you don't have access, please fill this out, and then, like I said, our access team will be happy to reach out and get a JIRA in place with your institution so you can access it, right? Assuming that you're part of, again, an academic, not-for-profit healthcare institute for now. We don't have a timeline about this, but, after this international expansion starts, we'll also presumably at some point in the future, no timelines, be also including, domestic commercial institutions as well. So yeah, Michael, probably right now you can't, but hopefully in the future you would be able to access this as a researcher in Nigeria. And that goes for other people in who are
Thank you very
much. Yeah.
Tara?
Yeah, I have a follow-up question.
Oh, sorry,
I
Michael.
wanted
Please continue.
Oh, thank you very much. I wanted to know how can I get to know any other webinar that I can get to learn about the use of all of us data apart from this?
You mean who's actually using, like, as far as researchers go? Sorry, maybe I didn't. Would you mind repeating your question again, Michael? Sorry, maybe I misunderstood.
Oh yeah, my question is that I want to get more familiar with the all of us data and do some research. So I want to know how I can get to follow up on webinar on how to use the data.
Yeah.
So,
okay, that's a great question. So if you don't have access and you want to learn about the data, I'd probably recommend going to this researchallofus.org page and using the data browser, which Michael described. And basically, this is aggregate public information about the data set. So all the concepts and variables that you can analyze in the Workbench are all available here, except it's not participant level data, it's aggregate level data. So for example, again, if you wanted to, just using HIV as an example, since the question was asked about it, if you want to find out how many, the types of, things that you can analyze about HIV in the Workbench or in the available participant data, you could find it here first. And then, if you want to research it more, obviously go to the Workbench, but this might be sufficient for some people, right? Just having this data is really useful, and it's available to anyone. And it's just on this researchallofus.org site. This is a public site. The user support hub is also a public site, so you can always go to that by clicking, and you can find that on the researchallofus.org site too. Let me just show you that. You can click the support button. I wouldn't, even though it's available, I generally wouldn't recommend clicking this unless you actually have Workbench access or plan to in the very near future, because a lot of this is geared towards using the Workbench. However, there is some information too about how the data is organized that might be useful. So if you are going to check it out, I'd check out Getting Started, right? Because this tells you types of all of us data, how they're organized, information about the OMOP vocabulary, which is probably useful for people. But so these sites are also public, and you can analyze, you can sort of learn about the data set there, how to use it.
I have two questions, if I may.
Yeah, please.
Yeah. One is regarding access, and the other one regarding that data set creation. So regarding access, I tried to register right now, and it says that my organization, Nova Southeastern University, has limited the number of people or something like this. So it says the institution has authorized access only to select members. How does that work? Is it limited by the university or?
Yeah, so yes, and you know, Cassie, my colleague, is on. So Cassie, please correct me if I'm wrong if I say anything correctly. She's our access expert, among other things. Basically, most institutions just have sort of a general, you know, allow all their users or all their associated people to have either registered and or controlled tier access. There are some institutions where basically they have to instead of a blanket sort of access, they have to you know, approve it themselves for individual researchers. I mean, you should be able to. It just probably will take a little bit longer to get them to.
Yeah. I just completed a second page for my institution. So.
Okay. Yeah, and that's, you know, that's just something that the institutions decide themselves. That's not something we impose on them in case people are curious. So, okay, and Cassie put some information there about Nova Southeastern. Thanks, Cassie. So yeah, that's a decision that the institution can make versus us making for them. So in case anyone's curious, but that's a good question, and I didn't mention that because it's kind of a unique case, but it does people do fall into that. So thanks for bringing it up. Yes.
So if my students want to access, they will have to go through the same process, register, and so on.
Yeah, exactly. So every person for themselves will have to go through the registration process. Yes.
Yeah. Okay, my second question is regarding the data set creation that you just explained. So if I want to say, I'm going to give you an example. Say I want to have a data set of individuals who are suffering from depression, and also they have raised inflammatory markers. So, sorry, I want to correct myself. Individuals who have depression and all the individuals who may have depression or not, but they have raised inflammatory markers. So it's and.
Yeah, I mean,
yes. Of depressive patients.
Yeah, and you could do that. So the depression, basically, I think in this case, the answer is yes. It would just have to be, you know, figuring out, number one, how you're classifying depression. Is it self-reported? Is it something that's in a EHR record, right? But if you, or both, right? But you can do that through the concurrent dataset builder, right? You can select which type of, you know, what types of questions you might want to include as being, you know, someone who is experiencing depression, right? And then for the other thing, for the inflammatory markers, as long as there was some kind of lab associated with them or condition code, you would be able to find it, right? So if you had one in mind, I'm sure as long as it was part of a person's EHR record, you'd be able to find that here. And you could, again, you could use the AND criteria. So, you know, you could, I'll just add that. So basically everyone. So in this case, you want to click and you'd want to add the criteria here as something else, right? And then you could do that. But so basically the short answer is yes, and the long answer is it would depend on how you're classifying these things. right? You have to sort of probably want to be careful and thoughtful about how you do that. But yeah, the data is all there for you to actually analyze. So yes.
Yeah, yes. So this, I just want to say that I'm looking for depression, and I'm looking for individuals with or without depression with raised inflammatory markers, and it is possible to join them together into one.
Oh, it's possible to join them. Yes, you can do that. You can do that in a notebook. Yeah. Sorry. Yep, you can join them if you want
Okay.
to.
Okay, thank you.
Yep.
All right, next, Rachel. And I know that we're pretty much almost out of time. So I see Rachel and Stefan. And if anyone else has any questions, we're happy to try to get those in as well. If you have questions, you can also put them in the chat.
Thank you. I was wondering, is geographic information available, like the state of residence that people live in?
Yes, good question. So for the registered tier, you have access to, you can find geographic data up to the state level, so the 2-digit zip code level. And if you have controlled tier access, you can find zip code data for, certain types of procedures and things, where it's basically where someone lives up to the 3-digit zip code level. So not really great resolution, but enough that I think it's still practical to work with, right? Especially in the control tier. And in case anyone's wondering, that's done to protect participant privacy. Why we have those sort of, why it only goes to the 2 and 3-digit zip code level. And thanks, Cassie. Cassie put in an article we have about that if you want to learn more about it.
Thank you.
Stefan?
Related, the depression example earlier was a good example of this. If we're just exploring the data to understand whether or not there's enough of a condition available for us to analyze, is there some documentation about where those categories come from? I imagine some could come from self-report surveys. For example, I was looking at the initial health survey that you have and some of the questions, whereas others might come from ICD codes, for example. And how do we know where the data are being pulled from as we're starting to explore the data? I understand later you may be able to join things.
Yeah, I mean, it's generally relatively easy to do if you're using the cohort builder or dataset builder, because like I said, it'll actually tell you like, okay, this category depression came up in survey data, condition data, or procedure data. So then you can go and then select, if I'm just clicking conditions, it'll only show the conditions, right? And I could select which ones I want to pick. And now I could go back and also add the other ones too if I wanted to. But it's pretty, when you're using these tools, it's pretty straightforward, but you can also find the concept IDs and SNOMED codes associated with them on the athena.org database, which will sort of give you information about what all those codes, where those codes are derived from and what they apply to, if that helps.
Thank you.
Yeah.
Any other questions?
One more, the geolocation, I can read the white paper that you've linked to, but is there, just briefly, are there broad definitions of urbanicity, those sort of categorizations available, or would researchers need to derive that if you have the appropriate level of access to data to zip code?
That's a great question. I'm 99% sure we don't offer that. And again, I think it's for participant privacy, but you could use, some kind of external database yourself and pull that information into the Workbench if you wanted to. Yeah, it's a good question.
And one thing I would say is that, the program is always really open to feedback. And so if there are things that you see as you explore, questions that you have, but also feedback, we'd love to hear from you. I think I put that email in the chat, but again, it's support@researchallofus.org. I see a question from Christopher.
Yeah, hi, thank you. Just a quick one. How does the Workbench handle longitudinal data? So say I built my cohort and want to pull all blood pressure measurements from electronic health records. Does it give me a longitudinal multiple rows per observation data set?
Yes. Yeah, the short answer to that is yes. And so like, as long as they As long as that record is somewhere in there, even if it's been done multiple times, it'll show up when you pull that data into the Jupyter Notebook for analysis.
Thank you.
So with regards to this question, how does it show up? So is it a new entry for different time points, or is it because I look at the data like in a data set rather than the database that is more complicated and layered. So for individual patient, will it appear in different time intervals?
Let me check something. I'm 99% sure. Yes, you can. That data should be there as long as, again, it was recorded in some way. Let me just go to the dataset builder. So for pick something here. Let's say for a lab or something, right? I'll just pick a random one. Probably body height's being checked by, more than once for people, I'm assuming, or actually maybe let's pick weight, because that's maybe even more likely to, or both, right? You should be able, if there's an associated date time, it should be an option. Let me show you what that looks like. Measurement date time. So that information, if available, would be, you could analyze it if you want, right? So that would be there in some way. You could use it to analyze something longitudinally.
So we are not able to see the data.
Well, you can. Well, you can. You just have to do so in a notebook, right, to actually do the analysis. You can see a preview table here, but if you want to actually list all the people in your cohort and have all that data available, you have to analyze it in a notebook, because it would require some kind of cloud analysis environment to sort of power that, because it's computationally intensive. And so even to a small degree, right? You need more than what these tools can actually show.
Any remaining questions? Okay. If not, thank you all so much for joining. Thank you for your patience. We really appreciate your sticking around and your interest. Again, please register for our newsletter. Please come to our office hours. Fridays office hours are open to the public, so you don't have to be a registered user to attend those. Please check out our YouTube channel, which has a lot of our office hour content there. So you can go see that, right now if you wanted to. And we hope that you will join our community of researchers. Have a great weekend.
Okay, hello everyone. I apologize again for that technical difficulty, but we're excited that you're here, and hopefully you'll get still some insight in this breakout session too. So I will go ahead and get started. My name is Sam Stewart. I'm a health services research analyst at the Data and Research Center. Specifically, Vanderbilt University Medical Center is my home here in Nashville, Tennessee. And so we're going to spend today's breakout session focused in the Jupyter Notebook, wrangling data. And if that's what you want to do here, that is what we're going to be focused on today. For those who are probably already curious, we will be using R today. We will not be going into Python, but if you do have specific questions, we can answer them. So I did want to have a few housekeeping notes before we get it started. So again, this presentation is intended to highlight the data in the researcher workbench, and will focus primarily in the Jupyter Notebook. If you are new to the researcher workbench, to include the data set, its structure, and the functionality of the researcher workbench, we highly encourage you to join Dr. Christopher Lord's breakout session 1, which is about becoming an All of Us research program user. It is a higher level, more introductory breakout room. So if you do want to go to that breakout room, just send me a private message and I will move you to that next room. I also want to make another disclaimer about our presentation today. We at the All of Us Research Program consider our participants' privacy the utmost importance. And so therefore, all the information that you're going to be seeing today is crafted from a DRC operational synthetic environment. And so none of the accounts or summary statistics represent real data. So therefore, if you see counts a little bit less than 20, it's because we're using synthetic data today. This data set is solely for education and training purposes and solely utilized by this DRC only. Therefore, researchers should not intend to reflect actual counts in their own research or Workbench accounts in the current mainline CDR. But this is a good example of how to do that in your workspace. I will make another final note. If you could just please hold all questions until the end of the presentation today. We do have some time set aside for us to have Q&A, so you can put your questions in the Q&A section or the chat, or kindly raise your hand during that time, and I will call on each person to ask their question. So I would like to introduce my colleague, Amon Kwame, who is a data scientist here at BUMC. She leads the data science and engineering efforts mostly on wearable device data, Fitbit data, and she's also involved in the back-end and front-end processes of the CDR, or the Curated Data Repository. Amon is a plethora of knowledge, and I have the opportunity to work with her daily, where she helps researchers figure out how to utilize the data set to their most efficient nature, helps researchers with their coding and any curation-related questions. So I'm glad that Amon is here today to help us learn more about wrangling data in the notebook. So here's our agenda. She's going to be doing a live demo, and she'll go into this just a bit more about what she's going to be doing. But she's going to be focusing on how to understand the data and summarizing it in your notebook, grouping, reshaping, and then joining and merging as well. And then when she finishes the live demo, her and I will take various questions related to what you saw in the demonstration, or just about the data set as a whole, or other tips that we could provide for you. So Amon, I will turn it over to you.
Thank you so much, Sam. Well, welcome everyone. Thanks for joining the session, and I hope that this will be helpful for you. I'm going to go ahead and share my screen, and this will be a Jupyter notebook. Okay, everyone can see my screen, right?
Yes, we can.
Okay, so like Sam mentioned, we'll be using synthetic data. So any row level data that you see or counts data that you see will not be real participants. Those are fake participants, but the data still has the shape and format of the real data, and the code that I'm using here is exactly the same that you would use to wrangle your data in the actual CDR. And so let's get started. So if you're new to data wrangling, you might feel overwhelmed with all the options that you have out there for packages, but you only need to know a few. And for data wrangling, tidyverse in R is the standard for all your methods and functions that really you need to wrangle any kind of data. So the first thing that we will do is to import our packages. So you just use library tidyverse. We'll also import big R query because this will help us to collect and read data from Google BigQuery into R. Since these two, SQL and R, are two different languages, we need them to be able to communicate together. So big R query is going to help us to do that. I took the liberty to collect some synthetic substance use data that we will use for wrangling. We won't have time to go through the explanation of the SQL query, but basically I have a few questions from the COPE survey related to drug use, also smoking, and also alcohol. I also have lifestyle survey questions for all these three areas as well. And then lastly, I collected some EHR substance use data. And then I have some demographic data just for some merging and wrangling. Now, one thing that I can advise you is if you're really new to R or data wrangling, find some good cheat sheet online. Just Google cheat sheet R. And this one, I'm going to use a lot of the the really short, succinct functions in this cheat sheet. And if you focus on mastering just a few of those, you can really work with any kind of data. Okay, let's get started. The first thing that you want to do when you're wrangling data is first you want to understand the data. So you want to have some kind of summarizing it so you can view the shape, the values in it, the dimensions, and that can help you understand what you need to do to get it into the format that you want. So here are just a few functions that most likely you will need to use when you're wrangling your data and when you want to see the data. So you can use view and whatever the name of your data frame is to view kind of the entire data frame. I mean, some of the rows will be cut off. This is good, or you can just type the name of a data frame. Now, I don't necessarily recommend this because if you have a large data frame, your notebook can crash. So better ways to do this would be to use the head or tail functions. So head and tail give you either the first n rows or last n rows. If you don't define an n, you will get the first 6 rows and the last 6 rows, but you can define n equals 2 and you get the first 2 rows, or you can do tail, whatever the name of the data frame is, 2, and you get the last 2 rows. So you can kind of see the values in it and the columns. You can also use glimpse. Glimpse is very helpful to give you like a compact display of the structure of a data frame, including the rows, the columns, the columns data types, and some of the values are in it. Dimension also gives you the rows and columns of a data frame. Unique is a function that will be very helpful if you want to see the unique value in a specific column. So if you do unique, whatever the data frame name is, and to get the column from a data frame, you have to use the $sign and then the name of the column, and then you get a unique value in whatever data frame. And distinct is very useful. I use it usually to count the number of unique participants in a data frame. And as you may or may not know, AOU data is long format, which means that a participant may have multiple rows of data. Each one is captured by timestamp, and it can be an event, a survey response. So if you want to count the number of rows of a data frame, you may not get the unique number of people in there because every person has multiple rows. So N distinct, and then you get the person ID from the data frame will give you the unique number of persons. And again, this is synthetic data, so these do not reflect the actual counts in the real CDR. Summarize is going to be your best friend for data wrangling. If you want to do group by, but you can use it on its own, and it really helps you to apply a specific summary function to a column or an entire data frame. So it could be any summary function. It could be sum, it could be average, anything. Here I'm just applying the n distinct function because I want to count the distinct answers from this data frame, and then you get to give the column that hosts the new count, you get to give it a name. Here I'm using summarize with across everything to apply the n distinct function to all the column in the data frame, so I don't have to do this individually. And then this really does the same thing as this. And notice I'm going a little fast, but save your questions and we'll answer them at the end. We only have 40 minutes for the entire presentation. And then count is a good way to count the rows in a data frame. So here I'm counting the rows in this substance use data frame. You can also stratify that count by column, and I'm stratifying it by this name and concept name. So I'm getting the number of rows for each of the concept names here. Now, this is a part that we're going to do live, starting from here. So subsetting data, grouping data, reshaping data, and joining data are going to be very important. You need to know these because you're going to use them probably 100% over your data wrangling. So for subsetting data, you either want to filter the rows of a data frame based on the condition, or you want to select columns, right? So let's start with filtering rows. Let's first of all show you, we want to filter rows of the data frame, the drug data frame, so COP drug DF. This is what it looks like. And let's say we want to filter all the rows for this particular person ID only. So let's give it a name. Let's call it COP. Oops. So what we're doing here, we're going to assign the new filter data frame, or the new data frame that we're going to get from the filtering to this data frame. So that's going to be the new name of it. And we just tap filter, whatever data frame that we want to filter from. And then you tap person ID. And then double equals. So if you don't put the double sign for equal, it won't work. And let's say we want this person ID. And then we're going to do head to see the results. And this is what we get. Now, let's say, and I'm not sure if you're familiar with Jupyter Notebook, but if you want to add a row, a cell, you click on the cell and then you click plus here, it'll add a new cell at the bottom. You can also move the cells up and down. And when the cell is running, there's an asterisk sign. When it's done, you see the number in there, okay? So now let's say we want to add two conditions. We want to filter multiple persons. So we want to get the data from multiple persons and add another condition to it, right? So we still do filter, COP drug DF. We can use person ID with this in, and then we create a list. So C in R will create a list. So C, and then we can put the person IDs in there that we want. So let's put 3 person ID here. And now we want to add another condition, right? We can just use N. And we want all the rows for these people where the survey data is after January 1st, 2020. And let's run. And this is what we have. So the same thing, because all of them are after January 1st. Now, oh, this is just a head, but let's see. Yeah, only these 3 people, these 3 rows. Now, you notice that here I used quotation marks and here I didn't use quotation marks, is because these are different data types. So remember, glimpse helps you to see the data type of a data frame. Let me show you. So for integers, you don't want to use quotation mark when you're doing the filtering, but for characters, you want to use. If you don't use the quotation marks for character, it's not going to work. And if you use them for integers, it's not going to work. Also, date times, you have to use integers with it. So that is a simple way to filter data. Now, let's go into selecting columns. So let's try to work with our alcohol data frame. And this is what we have. And I'm going to add a new cell. Let's say we want to select the person ID and the answer column. So we can give it another name, alcohol DF2. And then we do select our original data frame, what we want to get the columns from, or the data from. And we create a list with the column names that we want. So we want person ID and we want answer, right? And then let's just see the head, and we have our 2 columns. Now, sometimes it's easier to remove the columns that you don't want rather than select the column that you want, especially if you have a big data frame with a lot of columns and you only want to remove a couple of ones. So you basically do the same thing, but instead of C, you use -C, and then you put the columns that you don't want here, all the column that you don't want. So here, let's say we want everything but daytime. So we can put here survey daytime and then run it, and we get everything except the survey daytime. So filter to get rows, to filter rows, you use filter, and to select column, you use select. Now group by, you're gonna also need to know that 100%. This is where you can just group data in a data frame. You can count, you can do an average, you can do multiple operations by it, and you want to use group by and summarize together. And I'm going to show you how to do this. Now, I don't know if you've heard of pipes in R, but those are a way for you to chain multiple operations together to make your code look more concise, and it's easier actually to read. But I can show you, don't have to use your pipe, but I can show you the code with the pipe and without the pipe. Now, so let's say we want to work with, we want to do count in our smoke data frame. So let's look at it again. And we're gonna create a data frame for our count. Score. So we start with group by. Let's go by whatever data frame is it that we want to work with. And we want to group by question and answer. We want to see the count by person and answer, by question and answer. So we type question, answer, pretty straightforward. Now here it's not gonna do anything because we haven't said like which column to summarize by. So now we need to add another line of code with summarize. And then we have to use the previous data frame, the one that is grouped. And then you remember our summarize function, we can just define the new column name, and then we do n distinct person ID. And this is the count that we get. Now, how do you do this with the pipe? And let me show you the difference. So with the pipe, we start the same way, but then you put the data frame name, and I will explain what it's doing after I write it. So we do group by, question, answer, and then we put the pipe again, and then summarize NPIDs equals n distinct person ID. And summarize into, I forgot another. Oh, we got an I here. Okay, there you go. Now, what did I just do? With a pipe, you can just define the data frame once, and basically it's saying to R, apply all the functions after the pipes to this data frame in that order. So you only have to define it here. You notice that I didn't put it here, I didn't put it here. And you can also do this on multiple lines. Just make sure that the pipe is at the end of each line. And so if you want to apply multiple operations to the same data frame, you don't have to write multiple lines of code like this every time needed to feed the previous data frame. You can just do it all in a pipe. So you can use it or not use it. Now, the next thing is reshaping data. AOE data is long, like I said earlier. So sometimes our researchers want a way for them to view the data and have one row per participant. So let's say we're talking about survey data. They want one row per person, and the question would be the columns. So how do you do that? You want to use pivot wider. So pivot wider is actually quite simple. Let's just use our lifestyle DF. And let's say we want to change that, reshape it. So the values in questions become the column, and then the answers are the values in each individual cell. How do you do that? Let's call it lifestyle wider DF. And then pivot wider, the data frame that we want. And now, so this is a basic format. Pivot wider DF named from is the column that we want, whose value we want to become the new column. So that will be question, and then values from, that will be answer. Okay. So this is what we have. Let's look at our pivot data set. Let's look at the dimension here, right? Let's see how many people do we have here, and then we'll compare it to what we have now. So lifestyle DF, person ID, and then I also just want the regular dimension, like the number of rows and columns. So we got 226,000 people and then about 800,000 rows. So we know that's normal because each person has multiple rows. Now let's do the same thing here, but with our new data frame. So now the number of rows equals the number of unique persons. That means that our operation was successful. And notice I didn't do anything with person or survey data in the code, so those will remain the same way, and this is what I wanted. Now you can ignore this warning. It's basically telling you that it thinks that the rows are not unique, but as we just checked, they are, so you can just ignore that. Yeah, so this is basically how you do a pivot wider. Now, the next section, which is very important, I know I keep saying this, but I picked all the major things that you will come across in data wrangling, and merging is one of them. And I want you to really understand what's happening when you're doing a merge, like what does that mean, inner join, and so on and so forth. So we do have four major joins. You got inner join, outer join, left join, and right join. And there's one other join called an anti-join, which is pretty cool. And you'll see later what that does. So we'll start with an inner join. An inner join is basically, it's like the most common join. It's combining two tables based on the common column, right? And one moment, sorry. So the code is looking at the common column or common columns and see if there are any matching values in this column. And then if there are any, that's what it will return. And let me give you an example. And I will create a really small data frame so you can see visually what's happening after the join. I'm gonna use the Cope drug DF again. And here, arrange is helping me to sort the data by person ID. And then I'm gonna only get the first 5 rows of that sorted data frame, and I'm just gonna call it X, right? So X is this. And then we'll create Y, which is gonna be also an arranged version of the demographic, and with the first 5 rows. Okay, so this is the entire data frame. Now, if we do an inner join of X and Y, what's happening here? You only have 2 rows. So we have these rows here that match, and then this row here that match. If we had, if this person here had multiple rows, you would have all the multiple rows here, and then the values for that person will be attached, so it will be duplicated on each row. But here we only have one row for that person here and one row for that person here. So what's happening? You got basically the row here and the column, and then Y is attached at the end. Same thing for this person ID. So this is what's happening in an inner join. Now an outer join, basically this one, it combines two tables and keeps all the records from both tables, whether they're matching or not matching. And nulls will be inserted when they are non-matching. So let me show you. And now it's also the same as full join. So full join XY. So all the rows are included, and see it returns the same matching rows like it did for the inner join, and now it adds the ones that don't match and replace the parts where there's no match with nulls. Okay. Left join and right join function the same, but they're opposite essentially. So the left join will combine two tables by retaining all the records from the tables on the left, whether there's a match or not in Y, and on the table on the right, but only include the matching rows in Y. So all the rows in X and only the rows in Y that have a match in X. Again, let me show you what that looks like. So again, the inner join, we have those two match. So all the rows in X are here, all the rows in the survey, and then only the matching rows in the demographics are added, and then the rest is filled with NAs. Right join is the same logic, but it's the opposite. So all the rows in the tables on the right in Y will come, and then only the matching rows in X will come, and then everything else will be filled up with NAs. So right join. So we got all the tables, all the rows from the demographics, and then only the matching rows here, and then the rest is filled in with NA. Now, anti-join is another type of join from the tidyverse also, where it returns the rows in the first data frame that are not in the second data frame. So what are the rows in X that don't exist in Y? Or in this case, what are the rows in Y that don't exist in X? So let's see, anti-join XY. So all the rows in the survey data frame X that don't have a match for this person IDs in Y. And then if we did anti-join YX, then it would be all the rows in Y that don't have a match in X. Now you notice that I don't have to define here what join, for any of the join, you can write by and tell R which column you want it to consider as the common column, which is the common column. But because it's obvious, like R, we just look at the two data frame and be like, hey, you didn't tell me anything, but I see that person is the common column, so I'll join it by. And that's good. Typically that works, but sometimes you want to actually define the matching, like the columns that you want are to match on, otherwise your code might not work. What do I mean? So let me give you an example. Let's create another smaller data frame called Z, and that will be from the smoke data. And yeah, let me arrange it also. Arrange by person ID, and then get the first 5 rows, and that is Z. Now let's say I want to join X and Z, right? Inner join X and Z. Nothing. What happened? Let me show you what X is again. Because I didn't specify anything, Well, R is going to get all of the common columns and make the join. It's an and thing. So there has to be matching values in the person ID and the survey data and the question and the answer for one row for it to return that row. But these are two different survey questions. So the question will be different and the answer should be different, as they should be. So actually, this is not correct. So in this particular instance, it would be good. We want to specify I only want you to look at person ID to do the match, and then we're gonna get a return. Now, this is not the best thing visually. You can do several things. You can rename the columns, or what we advise is that you do as much wrangling that you can do on the data before merging it. So this next section isn't so much wrong merging, but like something that will make your data more visually comprehensible, because that's what you want at the end of the day. So here, let's try it out. If we had pivoted wider X and Z, what would have happened? Like, what would data look like, right? So let's try X wider. And if you remember our pivot wider function, X, and then actually I will remove the datetime column, and I'm doing it all in one code, so don't be confused. So again, I want to remove the survey datetime from X, and then now I'm gonna pivot that question value from answer, and this is X wider. Let's do Z wider. Same thing, I want to remove the survey datetime and widen it. Now let's see if we merge them here, like we did here, we'll see the difference in just how it looks and how easy it is to understand. So X wider, Z wider, and I don't even need to specify by, because now we only have one common column, and this is what it looks like, this compared to that. So that's why we recommend that you do as much wrangling on the data before you merge it together. Now, for the sake of time, I just went ahead and typed this in advance, and I basically widened all our data frames, and I join on the whole thing, so you can see how that would look. Here I'm joining the cope drug with the demographic, widening it. I'm doing the same here with the alcohol, and then here's a full join of those two data frames. And notice here, when you do the widening, the multiple answers are concatenated together with a comma. Now, it's not the same in Python. In Python, you would have to add another function to join the strings together, but pivot wider in R does it for you. And then here I created a controls data frame for the lifestyle. So everybody who has no for all of the answers, like no alcohol, no smoking, no drug use, I gathered here. So those are the controls. And then using the anti-join, then I can exclude everybody that is the rows are right here. Everybody, all the controls, I exclude them from this lifetime data frame. And then, so those will be your non-controls, and I just joined here using the pipe with the demographics. So this is an easy way to get like a control set and a non-control set. And then here I'm just doing basic count. So this is using the pipe. I'm selecting two columns, and I'm grouping by race and counting by unique persons. And I'm doing here the same with sex at birth, and I'm using the other pipe. They both work the same way. So this is the end of the wrangling, and I hope this is helpful, and feel free to ask any questions now.
Thank you, Amon, so much. Okay, So I will go ahead and open up the floor if you guys have specific questions for Amon about what we just learned in the demo, or if you have specific questions about the organization of the data set as a whole, this would be a great opportunity to ask those questions. I will note, because this is a public forum, if you are already a registered researcher of the Researcher Workbench, to just be sure not to show any participant or row level data if you are wanting to share your screen for any reason, etc., because this is recorded and will be utilized publicly later. So we don't have any questions in the chat, but if you feel more comfortable putting your questions in the chat, feel free and I'll read them aloud, or you can come off mute and ask your question.
I have a question.
Yes, Monique, go ahead.
Yes, thank you so much. That was great. I think you should teach. Do you teach?
Well, with AU sometimes, but not officially. Yeah, I've been hearing this a lot, so I'm thinking about it.
You should
Thank you.
teach. Yes, so in terms of the coding, because I'm relatively new to R, so in terms of the codes that can we have access to the codes that you have here?
Yes, we do have a very good data wrangling notebook workspace in our future workspaces that does go through some of these functions. And then are we able to share this, Sam? Do you know?
Yeah, I can share it.
Okay, yeah, we can share this workspace also.
Okay, great. Thank you so much.
You're welcome.
I also just put in the chat, Amon did another presentation very similar to this a couple months ago that's already uploaded on the user support hub for reference even as early as today. So while we get this demonstration prepared, you're more than welcome to view that one. Another thing is, like she mentioned, I also put in the chat the data wrangling featured workspace that does have a lot of the command line codes that she references. And then I will actually pull the Tidyverse cheat sheet she also recommended at the beginning, and I will also put that in the chat. Does anyone have any other questions?
Hi, my name's Kyle. I have a question not specifically on the coding itself, but more on the environment or the notebook once you've created that. I've got a couple that I've created where I have projects that are kind of on pause, and I get an email every month from all of us telling me that, it's whatever the fee is, $4 or $5 something a month. Is there any way to kind of freeze an environment or save just the cohort that you've created, I guess, so that you're not getting charged for it?
I think you're referencing, are you getting an email saying something about a persistent disk?
Yes.
Okay, I'm going to actually put this in the chat. So you can delete your environment and persistent disk at any time, as long as you save any intermediate files that you want to keep for later into your workspace bucket. And I'll also share a link to the article there as well. So my colleague Gage has a wonderful way of putting it, and I kind of steal it from him. If you are not keeping any of the files or outputs that you need for later, you can kind of turn the lights off at the end of the day, meaning delete your environment and delete the PD, and that will help mitigate any costs that you have associated with the PD. And that means that you are going to be actively saving things to the workspace bucket intentionally, and just kind of being aware of that. And that will help bring down the cost. So anytime you get that email, that's just saying, hey, you haven't used this PD in X amount of days. You can either delete it or just notify that you'll be charged that maximum amount of $480 for that month. Does that make sense?
Yeah,
And
so it's just like,
yeah.
so if I go to the delete environment that's up on the screen now. Like,
yeah. So if you click on here and like Sam say, make sure you save the files that you want to keep in your bucket. And then yeah, if you come here, you can delete the environment right here.
okay, great. Thank you for that.
On top of that too, I'll just add, you mentioned that you just wanted to save your cohort. So if that's truly all you want to save, is your cohort or data set, you can go ahead and even delete your notebooks and delete your workspace bucket, and that will cut down on cost even more. So up
Because
to you.
keeping the notebooks still has a
Yes, so the notebook is automatically saved to the workspace bucket. Although the storage cost is very minimal, it's like rounded up to 3 cents per gigabyte per month. If you don't want any storage costs associated with your workspace, then you would delete everything out of your workspace bucket as well as the notebooks, if you truly are just looking to save the cohort and data sets.
Okay, thank you for that.
There was also a question in the chat from, I believe, Wilson, asking if there was a summary of cost. So that cloud analysis console that Amon just pulled out, once you customize your environment, that'll show you or estimate how much it'll cost when active per hour or when paused. And then the article I shared also tells you a little bit more on the persistent disk cost. Outside of that, we don't have any parameters to help estimate the cost for every specific project, because it really does vary depending on how much data you're pulling in from BigQuery and how much analysis you're doing in your notebook, and then how much you're storing as well. So it'll all vary. However, we do have a user support hub article called Estimate How Much Your Project Will Cost, and it's based on other investigators in the DRC who have run analysis from start to finish and what it cost them in their environment for their specific amount of samples or participants. So I will share that as well. Another thing that I personally do is I always look at the beginning of the workspace for our featured workspaces or demonstration projects to see how much it costs for that individual to write that. Those things are available to anyone under the featured workspace section of the researcher workbench. So that'll give you more of an idea of how much a similar project will cost in your workspace. Any other questions about costs, storage, optimization? This would be a wonderful time as well to ask. Oh, Engage just shared an office hours about that information as well. Well, if anyone doesn't have any questions, I myself and Amon will stick around for the next few minutes, but we just want to say thank you for joining us today. I do want to highlight a few more resources at your disposal. So we did go over a lot of information, but we are still here to help. So our help desk support and our user support team, which I'm a part of, Amon's a part of, Gage, we're here to help researchers as they utilize the researcher workbench. So we have various opportunities for you to engage with us. You can always email us at support@researchallofus.org, and someone on our help desk team will hopefully be responding to you in 1 to 2 business days to get that initial inquiry started. So you can email us as many times as you need to. We do offer drop-in office hours every Tuesday at 1 p.m. Central Time. Myself and my colleague Gage help lead those, and members of our data science team, such as Amon, will also be present to answer any of your questions. Those office hours are closed to registered users of the Researcher Workbench, and you should receive an email to your institutional email address affiliated with your account to register for those. We also host bi-weekly Friday office hours, which are open to the public, that sometimes have members from the support team present on different topics around the Researcher Workbench, or we have guest speakers as well, highlighting their research. Anytime those are recorded, we then will upload them to our YouTube channel and on their user support hub. And then finally, mentioning the User Support Hub has all types of articles for you to reference, to include some code snippets there, and to see more about the features that we have of the Researcher Workbench. And then if you want to use that QR code, that will link you to our events calendar to let you know other events and webinars that we have available at your disposal. And so I do want to say thank you on behalf of the Data and Research Center and the NIH All of Us Research Program for participating in this webinar today. So if you are finished and you want to go about your day, you can leave the webinar from this breakout room. Otherwise, myself and my colleague Amon will stick around the next few minutes if anyone has any last-minute questions before we break. This was recorded along with the intro session and should be made available to you soon. So thank you all for joining. Feel free to drop off the call and we will stay for the next few minutes. Thank you so much.
Their visions and the summaries on 7 different angles, as shown in the bottom right corner, from big data to genomics, electronic health records, diversity, longitudinal cohorts. Those 7 aspects provide an amazing picture of, precision medicine in 2030, which is our dream. So this includes, of course, both opportunities and challenges. For instance, how can we better link, genomic data, electronic health records, and longitudinal cohorts to improve population health in diverse groups? So I think my research is quite inspired by that review paper. Next slide, please. So specific to genomic medicine research on substance use or mental health, here I would like to quote a few sentences from Dr. Jordan Smoller's review paper for psychiatric genetics. That is, all major psychiatric disorders have a familiar and a heritable component, and the genetic architecture of psychiatric disorders is highly polygenic and includes the full spectrum of DNA variations. So not just monogenic genes, but quite a few DNA mutations. Genetic discoveries are providing new windows onto the underlying biology of psychiatric disorders. And the genetic influence on psychiatric disorders is now transcending our clinical diagnosis boundaries. So with that knowledge learned, I also published a method review paper with my PhD mentor, Pak Sham, to cover a list of existing population cohorts, such as, all of us, UK Biobank, extra. Genetic approaches, statistic genetics, and the bioinformatics tools, and the omics database. So on top of those resources, we also review how previous studies use them to better understand schizophrenia, from genetic determinants to clinical classification and the risk prediction for that disease. Next, please. So my current research is focusing on opioid use disorder and suicidal behavior, still in the Department of Psychiatry. So this snapshot shows the overall analytic framework to study them in my lab. So we have collected existing genomics data in 3 different tiers. The primary tier 1 data is raw phenotypic and genotypic data from all of us, UK Biobank, and the Kaiser Research Bank. In total, they cover around 70% European population and 30% other minority populations like Black, Hispanic, Asian, and extra. So tier 2 data is mainly GWAS summary statistics from different consortium or databases. For instance, Psychiatric Genomics Consortium, short for PGC, and the Million Veteran Program, short for MVP. So this disease-specific GWAS covers not just opioid use disorder and suicide behavior, but also many other related substance use traits or mental health conditions. Tier 3 data is for functional omics data, for instance, from the raw data from the GTEx version 8, version 9, and the Psyche encode. So this data covering epigenomic and the transcriptomic data for brain tissues and the different cells. And the samples coming from both European and non-European. So we then combine these three tiers of data together for multiple genetic epidemiology and integrative analysis. For instance, typical post-GWAS meta-analysis and annotation, like in terms of the gene function, clinic risk scoring with multi-view clinic data, genomic risk scoring with both common and rare genetic variants, thanks to the whole genome sequencing data, and the risk model training and the validation, using both retrospective and prospective data in the big cohort. Next, please. So in the following few slides, I will share three related analyses for opioid use disorder, leveraging all of us data itself, or the combination of all of us and other external data sets. The first one is to identify social demographic correlates of opioid use and use disorder in all of us. I think Dr. Michael Lyons cited this paper in his slides. Thank you very much. So this is a demonstration project led by my colleague, Dr. Xuehan Ye, whose office is just the next door to mine. So I have to say I contribute only minor to this paper, and I would like to give full credit to Xuehan and the whole team. In this study, we included total 214,000 around subjects from all of us version 4 dataset. So all of us now releasing, I think, version 7. So it's like this is analysis performed around 2 years ago. So the participants were included if they gave response to lifestyle survey questionnaire or had opioid use related electronic health records before. So the survey covers questions for lifetime opioid use and is used in past 3 months when they were included in the program. So EHR covers a few different domains like drug exposure, like morphine, codeine, those kind of opioid medications, and condition table for diagnosis. Descriptive table and a logistic regression model were used to analyze this epidemiology data. Next, please. So I want to thank again to Dr. Michael Lyons. So he showed the descriptive table of this study in his presentation early this afternoon. So here I show another table in that paper. So it's the odds ratio and the confidence interval of 4 different social demographic variables from the multivariable logistic regression. So this includes gender, age, race, ethnicity, and the country of origin. So from this study, we found lifetime prevalence of street opioid use is 4%, non-medical use of prescription use is 9%, and 40% participants received prescription opioid, and 1% of them had diagnosis of opioid use disorder in the past 3 months. So in addition, so when comparing to other reference groups, we found men, younger age, white population, and US-born participants are more likely to have opioid use disorder in that data set. Next, please. So I did some extra exploratory analysis to compare the demographic patterns of opioid use between all of us and the UK biobank participants. I know a lot of the audience have more knowledge on the UK biobank. So as you can see from this table, both opioid use disorder patients, so case are patients, and the regular opioid use subjects, so they are opioid exposed controls. They are not general control. They are, they have to be exposed to regular use of opioid. So we found, both opioid use disorder and its matched controls are more prevalent in US than in UK. So this may be due to the population structure and prescription policy, quite a few country level reason. So in addition, all of us has more sample and the data for analyzing non-white diverse populations. As you know, less than 10% of subjects in UK Webben are from non-European ancestral background. Next, please. Okay, so the second analysis is to perform cloud-based genomic analysis for opioid use disorder. So we started to implement genetic tools and pipelines since I think early 2022, when version 5 control T data became available to its registered users. Up to now, our group has performed 5 different kinds of analysis with tools available on the cloud itself or newly installed by ourselves. So this covers quality control steps, say using HAIL to manipulate big matrix table, genome-wide association analysis by P-Link, and or the R-Genesis package, rare variant association test by the STAR package developed by the Harvard group, polygenic risk scoring by PRSCS2 and PRSCS-X in the diverse populations. And another angle is my special interest in HLA. So we call HLA haplotype from the CRAM and BAM file of the whole genome sequencing using HLA LA and the Karami software. So we run these tools in 3 different job modes. They are complementary to each other. Sometimes you test the jobs by interactive Jupyter notebook or the command line terminal. They have like just in the traditional cluster, the command line tool. And the Widow is a language for submitting your batch job to all of us. Next, please. So here are snapshots of different quality control steps in regular GWAS analysis, including checking of sample missing rate, heterozygosity rate, global ancestry for mixed populations, say Black African Americans or Hispanic Americans. Scatter plots for top principal components, examination of relatedness between individuals, and the sex mismatch. So a lot of these quality metrics are already provided as auxiliary data on the All of Us cloud. For instance, the genetic ancestry percentage, and also the the classification of which genetic ancestry population from. So, and the top 20 principal components, pairwise kinship matrix, and a list of related samples created in the cloud. So you can directly import them into your own workspace without the need to calculate by yourself. Sometimes it's time consuming, but this is very convenient. Next, please. So this table shows top SNP association from our preliminary GWAS on opioid use disorder in all of our samples. By the way, so when we run the analysis, the samples are from the SNP array, not from the whole genome sequencing, because at that time point, SNP array have more, it's a larger sample size than the whole genome sequencing data. So as you see here, the most significant p-value is still above like 5 times 10 to negative 8. So no genome-wide significant hit was found from this all of us discovery analysis in either white or Black population. We further checked this list of suggestive SNPs in summer statistics of UK Webben or MVP, but we did not find much consistency in terms of the significance. And a lot of them are not present because the array difference. So data harmonization is needed to compare SNP level association across these 3 cohorts. And after that quality control, we aim to perform a cross-cohort multi-ethnic meta-analysis to identify some genetic biomarker for OUD. Next, please. So the third analysis I want to talk is the genetic relationship between opioid use disorder and suicide attempts. So there might be a jump here, like why study OUD and suicide together. Here I would like to highlight a few reasons So deaths from opioid misuse and suicide attempts are both increasing, and there are high rates of occurrence of OUD and suicide, especially in mental health patients. However, it is hard to estimate OUD and suicide correlation and causation using epidemiology data only. And it's also unclear how their genetic liability relate to each other. More importantly, via cross-phenotype genetic analysis, so we may be able to design some mechanism-based preventive strategy that may work for both conditions. Next, please. So here's a published work by my group in this year. All data included is not from all of us project, but more traditionally using UK Biobank and the GWAS summary data from multiple resources. The figure shows the overall flow chart for our analysis for on both conditions together. These include clinic association, genetic association, and the Mendelian randomization analysis. So we also control for genetic correlation from other psychiatric disease. For instance, alcohol use disorder, bipolar disorder, major depressive disorder, schizophrenia, anxiety, and PTSD. However, only samples with European ancestry were included. Next, please. From that analysis, we found heritability of OUD was genetically correlated with suicide attempt, even after conditioning out the polygenic effects of major psychiatric disease. And the combination of AMR analysis suggests a possible causal association from genetic liability for suicide attempt to opioid use disorder. However, there are a few remaining questions to be answered. Can you click the slides? Okay, for instance, opioid use disorder is a suicide pattern of in non-European population is unaddressed. Pleiotropic loci and specific genes are not fully explored, and the role of chronic pain is not well considered. And lastly, the temporal development of the onset and co-occurrence is still unknown. Next, please. So then let's see what all of us data can add new sites for their relationship. From demographics data, so this is just like a baseline enrollment. So we found patients diagnosed with OUD has a higher likelihood of committing suicide attempt in overall sample, and also in those samples without major psychiatric disease. Besides white population of UK Biobank and all of us, this pattern also generalized to Black population in all of us. Next, please. So we further check the COPE survey data in all of us. So this is a longitudinal multiple tampon survey conducted in the pandemic. So we use PHQ-9, item 9 response to estimate the suicide source. So in total, 6 tampons were collected, spanning from May 2020 to February 2021. We then linked a pre and a post-pandemic psychiatric disease status. So they are overall status and also specific to opioid use disorder. And then link it with suicide source cross-sectionally, and it's a change of suicide source across pandemic. For cross-sectional analysis, we found psychiatric disease overall status, and also OUD itself is associated with higher rate of suicide ideation for each tampon. And when examining the whole longitudinal data together, we found suicidal ideation rates significantly dropped from earlier to later pandemic months. But the interaction of the psychiatric disease status or the OUD status and the time on the trajectory of the thought is not significant. Next, please. Another new hint is from the genomics data from all of us. So we perform a similar polygenic risk score association between opioid use disorder and suicide in both white and the black population. The training data of GWAS summary statistic is from the MVP, they published latest OUD or the suicide source and attempt GWAS, I think last year and this year. So the target data is from all of us. There is no sample overlap between these two data sources, as you can see from this table. We found cross-phenotype polygenic association, for instance, from the MVP OUD to all of our suicide attempt, or from the MVP suicide ideation and behavior to all of us OUD. So these pairs are significant in white, but not in black. However, when we check the intra-phenotypes polygenic score, which means OUD in MVP to OUD in all of us, or the suicide in MVP and suicide in all of us, it kind of show the curve for black population maybe not adequate. Next, please. Okay, so I share a few preliminary evidence from analyzing data. So I hope, hopefully I'm convincing you that, the usefulness of the all of us data. And the take-home messages is, all of us research program provides abundance of phenotypic and genotypic data for genomic research on substance use and its related mental health conditions. So all of us cloud platform enables efficient genetic epidemiology analysis for its own and combine data with external sources. The integration of all of us and non all of us facilitates my genetic research on opioid use disorder and its connection to suicide behavior among diverse population. So our preliminary results show OUD prevalence difference between US and UK. But still negative findings for OUD GWAS within all of us itself. But we have some significance of polygenic association when we perform cross-cohort analysis. Lastly, next slide. So lastly, I would like to thank a lot of my colleagues at Henry Ford Health. So as the regional medical center for all of us, Henry Ford have a big group to collecting new samples and data. And also we have a big research group to leverage this data for substance use and mental health research. So I would like to especially thank, Shehan, Lisa, Brian, and a few other colleagues. Special thanks to the data providers, UK Biobank, MVP, all of us, PGC. And lastly, I want to acknowledge my funding sources. Thank you very much for your attention.
Fantastic. Thank you so much, Dr. Gui. We will move on to Jennifer's section. We have about 12 minutes left in the scheduled time. We did have those technical issues, so we apologize for kind of running a little bit behind schedule. If you need to drop off early, very much understand that, but we will move on to Jennifer's section.
Oh, thank you, Michael. So after Dr. Gui's amazing and comprehensive study, I will give a very simple demo on how to connect phenotypic data and genomic data in the research workbench. So we will cover how to query the phenotypic data stored in BigQuery using a very simple and free to use tool, which is called Cohort and Dataset Builder. And we will cover how to get and load the genomic data in the research workbench, and then show how to connect these two data types. Next slide, please. Yes, so the phenotypic data of interest today is the social determinants of health data. It is survey data with 80 questions, as Michael mentioned. It's covering participants' neighborhood, social life, stress, and the feelings about everyday life. For today, we are only interested in we focus on the social isolation. And for the genomic data, there are various types of genomic data in the research workbench. Today, we will use the short variants, also known as the SNPs and indels, in the short-read whole genome sequencing data in the format of a Hill matrix table. And after linking these two data types, we will run a straightforward GWAS to identify the variants that are associated with the susceptibility to loneliness. Last slide, please. During the demo in the research workbench, to comply with the data and statistics dissemination policy, I won't show any row level data, which is also sample level data, or I won't show any variants with allele counts less than 40, and all the aggregate data shown in the demo will have sample counts greater than 20. So with that being said, let's, I think I need to share my screen. So.
All you, Jennifer.
Yes, good. So share. So I think you should be see the research workbench. If not, please let me know. So
you're good to go.
First, okay, great. So first we will visit the cohort builder to create the cohort of interest, which are the participants with both the social determinants of health data, edit and finish, save, and also the short read whole genome sequencing data. Now we can see that there are around 70,000 participants in this cohort. We create this cohort and name it as social determinant of health, short read whole genome sequencing data, and test. And save it. Next, we use the dataset builder to create a dataset. So we select the cohort we just created and select the social determinants of health and select the fields we want. That is, we want the person ID, survey type, question, and answer. And we save this dataset. We name it as social determinants of health and short read WGS. Okay. And we save it. So next we can analyze this dataset. So this basically generates the SQL code to retrieve the data we specified in the dataset builder. The code we can export to a notebook, or we can just copy this code for analyze. Since I've already have a notebook for this, then I would just go ahead to the notebook I've created before this demo. So this is the code generated from the SQL code generated from the dataset builder. We can see that if you run this code, you will see that it takes less than 3 seconds to retrieve all the social determinants of health data for the cohort around 70,000 participants. And the first part of this notebook is just to import some packages needed and to record time and cost to run this notebook. I'm using a default tail analysis environment. The cost rate is like $0.73 per hour. The data retrieved from the database using after running this cell is saved to the notebook in the type in the format of a data frame. Now we can see that all the 4 fields we specified in the dataset builder are in this data frame. So, and to comply with the dataset data and statistics dissemination policy, I remove the first column, person ID, to show you how the data are present in the data frame. And so as I mentioned before today, we are only interested in the social isolation. The question, and there are 80 questions in this data. So the question we are interested in that is associated with the social isolation is how often do you feel isolated from others? Let me zoom in so you can see clear. So now we filter this data frame to the question and remove the rows that do not have the answer. So which leaves us to around 67,000 records. And normally, we will transfer the text values to numeric numbers for downstream analysis. So we can see that is successfully transferred. And now we can save this phenotypic data we just retrieved and cleaned a little bit to a TSV file and copy it to a workspace bucket, which is a permanent storage for later use. So we just retrieved the data, the phenotype data, and save it to a permanent place. So now we can use the environmental variable to get the path of the genomic data and use Hail to load this genomic data. And we can see that there are around more than 1 million variants in this data set and more than 245 participants in this genomic data set. And I filtered this genomic data to a specific region just for demonstration purpose so that all the variants here have allele counts greater than 40. So this, for the variant information, we have the position of the variant, the reference alleles, alternative alleles, the filter status, and some essential information like the variant QC and the info fields like allele counts, allele frequency, and allele number. And now we have load the genomic data into Hail, and we can load the phenotypic data we just saved into Hail as well, and we use this function, and then link these two data types together into Hail. Use the field person ID, which is consistent across different data types, and we can see that we have successfully added the phenotypic data into the genomic data. Like in the new matrix table, besides the person ID in the old matrix table, the 3 fields that are in the phenotype data already in the new matrix table. And now we have a data set with containing both the phenotype data and the genomic data. We can build a model to run the GWAS. That is, we run a linear regression, and we can save the results the same to your workspace bucket, which is a permanent storage. And we can also visualize your result and save your result to your bucket for later reference. So the last is just to show that it takes less than 9 minutes to run this whole notebook and cost around 1 cent. So that concludes my demonstration, and thanks for your time. I will pass back to Michael.
Thank you so much, Jennifer. Let me just go back to here. So we are low on time. We have about 2 minutes left in the scheduled meeting. So we can open this now up to any questions. And before we do that, I really want to thank Dr. Gui and Jennifer for coming out and giving those fantastic talks. That was wonderful to have you both here. So thank you for that. And if there's any questions, feel free to jump off mute or throw them in the chat. I see a question of where can we find this Jupyter Notebook file for integrating the data? Jennifer, do you want to dive into that?
Yeah, so you need to create your own notebook to do this. So every time you log into the research workbench, it will ask you to create a workspace. And when you create the workspace, in the workspace, you can create a notebook. So I think Chris Lord, the session 1 will cover this part. That is the very first step after you have the account for the research workbench. Yeah, you can. I think you'll get more information from watching the recording for the session 1, probably.
We also have a wonderful support hub that is available to the public where you can kind of go into and read different articles. We also have office hours and new user orientations. The calendar is on the support hub. So if you're curious about kind of learning more about the program or how to do that, we have a wonderful support staff and a lot of materials to look into to help you all learn this system and how to go through it, because it can be overwhelming and there definitely is a learning curve. But we want all the researchers to succeed and run the analysis that they want to. So yeah.
I want to address Dr. Johnson Pollock's question on defining opioid use disorder. I sent a short response in the chat box. So there are quite a few sources of data to define opioid use disorder in all of us. They have the so-called mental health questionnaire, like have you ever been diagnosed with psychiatric disease, and then more specific to opioid use disorder disease or other depression disease. Then there are like EHR data, ICD-9, ICD-10, SNOMED diagnostic codes for OUD. So in our group, we also try to extend the sample size a little bit by using medication records in the EHR. But this definition is a little bit of risky, given the medications are prescribed for not just OUD but also chronic pain and even depression, say for the buprenorphine.
Any additional questions? Right. Well, oh, I see something pop up in chat. Oh, perfect. Thank you. Yes, I think with that, we can close it out. Thank you all so much for coming and learning more about the All of Us research program. We really hope that you can utilize this resource in your research and really make a positive impact. And we're looking forward to seeing what you all do. And thank you all again for coming.
Thank you. Bye.